Hello and welcome to ThePrint. My name is Radifah Kabir, and today we will be interviewing a very special guest, Dr. Pragya Dhruv Yadav. Hello, ma'am. Welcome to ThePrint, and thank you so much for joining us.
Good morning, Radifah. Thank you so much for inviting me today.
Good morning, ma'am. Dr. Pragya Dhruv Yadav has received a Vigyan Yuva Award in Medicine this year. Congratulations, ma'am, on winning the award.
Thank you very much.
Now, before we begin the interview, I would like to tell you a bit about Dr. Yadav. Now, Dr. Yadav is a senior scientist at the National Institute of Virology, which is also known as the NIV. It is located in Pune and is India's research institute in virology. It is a part of the Indian Council of Medical Research. Dr. Yadav has made significant contributions to the fields of communicable diseases and understanding viruses. These include Zika virus, Ebola virus, Marburg virus, Mpox, and also SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Virus 2, or the novel coronavirus. This was responsible for the COVID-19 pandemic. You will be surprised to know that Dr. Yadav is among the first scientists who had detected the first COVID-19 cases in India. She also had a very important role to play in the development of the COVID-19 vaccine, Covaxin, which was India's first indigenously developed COVID-19 vaccine. Now we would like to begin with the interview. So, Dr. Yadav, please tell us a bit about your work, about the nature of your work, and which research won you the Vigyan Yuva Award.
Thank you very much, Shadia, for introduction and your appreciative word. I'm very happy that Government of India is giving me award, and I will be the first one from ICMR to receive it. So as you introduced, that I work in ICMR National Institute of Virology, Pune, from last 24 years, and I did my PhD. So I was trained in virology. And when I joined, during the first year, we could see the outbreak of SARS coronavirus, which killed thousands of the people across the world. At that time, we realized that India lacked biocontainment facility and also the infrastructure for testing the high risk group of pathogens. So, so that was a kind of alarming. And fortunately, I received a fellowship from a Center for Disease Control USA, and ICMR sent me on deputation to get trained in that. And after coming back, I started developing different diagnoses for high risk group of pathogen. In the meantime, India has its first BSL-3 facility in ICMR National Institute of Virology that help us in starting the research in high risk group of pathogens. And the first, which I started with Cassino forest disease, is a tick-borne disease, flaviviruses, and cause a viral hemorrhagic fever located in Western Ghat of the country. The first thing was to develop a test and then do the research to understand more about the disease. So we started that, and now we have 12 laboratory across the Western Ghat who can do the daily testing of this pathogen. We are working on a different aspect of this virus. Then, as you know, the 21st century is a being of emerging diseases. So there was a number of public health issues which arrived, and we came across and we answered the questions through different research programs and projects supported by fund of Government of India.
Yes, ma'am. That is so inspiring. And as you said, you have laboratories in the NIV. So can you please tell us what role you play in the laboratory you're heading? What research is conducted in that lab? Also, can you please tell us what containment laboratories are? Because I read somewhere that you also handle containment laboratories. So please tell us about containment laboratories and also about the labs you're heading.
So the BSL-4 lab, which I'm heading, was established in 2012. It was validated and become functional in 2013. And being head of this facility, our main responsibility was to prepare not only the diagnosis assay, but also the vaccine and the research on high-risk group of pathogens. And as you know, after 2013, we have seen many outbreaks. As we discovered Crimean Congo hemorrhagic fever in 2011, and then we did develop many diagnostic assay, which was also commercialized. But we also did a study to find out what are the reservoir, how it is transmitting, and what we can do more to understand about the disease. Then further, we could see the outbreak of Ebola in country, and then you all remember the Nipah outbreak, which happened in Kerala in 2018, 19, 21. Recently, there was an outbreak, and we enhance our capacity in a way that not only we can detect, but we also can help to control, maybe saving the life by early containment program with the state government and central government of India. During the COVID pandemic, you all remember in 2020 when it started, there was a lot of panic that how we are going to do it. And with our National Influenza Center, which is our one of the laboratory who work exclusively on respiratory pathogen, we developed the assays and we trained the VRDL facility, which is a Department of Health Research laboratories, a very mega project. Now we have 158 laboratory across the country who can do viral diagnosis. So after the training them and initially hand holding, we then move on working on antivirus, working on the vaccine. And as you know, all know that success story of Covaxin and then Zydus DNA vaccine, which came up later. Then we participated in different studies and answering many questions regarding the vaccine efficacy, neutralizing capacity against different variant of concern. And then once a variant of concern emerge during different waves, we also work on finding out if the variant of concern has any role in mucormycosis or in other reinfections, breakthrough infection. And so these 3 years during the pandemic initially was focusing on research-based question, answering through the research, through very ethical practices. So there are a lot to talk about it, but in summary, I can see with my team and with the support of higher leadership from ICMR and Government of India, we could not only do the research, but in the pandemic, there was a need to publish all the data very quickly as soon as possible because everybody was working in the same direction and your research input can help others to invest in to get the answer very quickly. So we publish a large number of papers on COVID, but we continuously focus on so publication. Why I'm talking about the publication, because research data help others to develop the products, do the better research. And in the COVID, actually everybody was reading other international papers, so it was very important to publish, put on the public forum to showcase and also showcase the India's research capacity. So we published around 186 papers on COVID, and we could produce those data, share with the policy maker, and then help India's policy in COVID as well.
Yes, ma'am. Yes, it must have been very difficult to publish so many research papers in such a short while.
It was, but I think this was an opportunity being a virologist for not only for me but for entire NIV team and our team to serve the country by research program. As we all know that we can serve the country in many ways. Some people go for war, some people do the engineering, marvelous buildings, infrastructure, and being a scientist in the public health research, you can always do something which can help people to have a better life. And I think the COVID was an opportunity to showcase this strength for the country and entire team of NIV. And I was one of the member of the big team. We could demonstrate that.
Yes, ma'am. We should all thank you for your commendable work because had scientists not worked fast on the COVID-19 strains to understand their working, vaccines wouldn't have been developed. So, thank you from the entire scientific community. Thank you from the entire country for all your contributions.
Thank you very much. And once again, I would like to thank Government of India and Principal Scientific Advisor for choosing me for this award, which is very motivating for the young generation and for the young woman in the country to participate in the scientific study in future.
Yes, ma'am. Ma'am, my next question to you is, you also decode virus strains, especially when there is an outbreak, such as that of Ebola, Zika, Nipah, or Mpox, for instance. So please explain, how do you decode the strains of viruses, especially when there's an outbreak? Can you please explain the steps in simple language?
Yeah, so whenever there is an outbreak, what we mean by outbreak, whenever there is a case of any disease which is unknown or known, and if you would like to know what is the causative agent, so we test by different diagnoses, say we call serological method, molecular methods, and real-time PCR, RT-PCR, some of the name which you all have heard during the COVID pandemic. So then if we detect the pathogen, then there is another method to call the propagate the virus or isolate the virus in a more in the laboratory using the animal model or cell culture, and then we can use that virus stock for doing the more studies, developing assays, developing vaccine as we did for Covaxin in the inactivated virus vaccine. But we can also use the virus for developing different assay which can be utilized for testing. Moreover, if it is an unknown pathogen, so now there is a new technology come, and in the past it was used to call Sanger sequencing, but it's called now next generation sequencing, where you can decode the nucleotide of each pathogen present in the samples, human or animal samples, or in a vector sample like mosquitoes, and you can read them using the bioinformatics tool and identify what pathogen is present. So these are some of the modern tools which was not there in the past, but we have. And with the COVID pandemic, these tools has been proliferated and now present in India in many large number of the facility. So in a way, actually, these technologies has helped to enhance the response not only in India but globally to detect and predict the disease discovery.
Yes, ma'am. So can you please tell us how you decoded the first three COVID-19 cases during the pandemic?
So when the COVID started, that time we didn't have any COVID specific test, but we do had the nested RT-PCR, which was used for the other coronaviruses detection. But at the same time, we had a very beautiful tool of next generation sequencing, which I was describing. And with the help of these two, and the later on, even WHO shared the sequences of primer sequences of coronavirus to detect. So we could also get it that, and using the real-time PCR, RT-PCR, and next generation sequencing, we could detect the initial cases. But prior to those detection, we screened a large number of the samples, and they were negative. So it was a constant efforts to detect the cases. And initially, if you remember, the guideline was to the people who are coming from abroad. So and if they have any symptoms, and if they have showing, then those samples should be detected. So we followed with all the people who were coming from abroad, and if they showed any symptom, they were detected. Most of the people turned to be positive for other respiratory infection, but the medical student who arrived from Wuhan, they turned to be positive for SARS coronavirus. But that was just beginning, and then later on, we could see more cases in March. And if you remember, there was a mission which was sent to Iran for the standard citizen. Then we expanded the facility, and now with the efforts of Department of Health Research, we have more than 3,000 laboratory across the country who can do the testing. But later on, we also developed TrueNet PCR assay, which can be utilized in small laboratories, in district level facilities, very cheaper cost, and that was also expanded across the country to enhance our testing response.
Ma'am, in the NIV, there are certain containment laboratories. Are these meant to store certain viruses?
So in NIV, we have BSL-3 and BSL-4 facility, and we work in BSL-4 laboratory as a biocontainment, where we also store the highly infectious disease pathogens, and that can be utilized for different research whenever there is a threat, or if you would like to make a vaccine, want to do a preclinical studies, neutralization studies to understand more about these diseases.
Yes, ma'am. And please tell us what your role was in testing Covaxin, in testing its efficacy, in developing it, and all other stages of vaccine development. What was your role in it?
So, as you must have seen the movie, The Vaccine War, which was based on the Covaxin story. And in the beginning, when in the virology section, whenever there is an infectious disease emerge, which is unknown, the first thing which we do, like identify the virus, but then similarly you propagate the virus, do the virus isolation. So as soon as we receive these three earlier samples which turned to be positive, we try to start the virus isolation process. But as it was a new pathogen, nothing was known, and we failed miserably. We tried different cell cultures, different animal models, and the why we were doing, because we wanted to have a vaccine from the beginning, because it was a new threat and nobody was knowing what is going to happen. So but 2 months like continuously we try with the earlier sample, it didn't work, and we analyze what is going wrong. And based on that experience, we as soon as the March the cases happened in with the Italian tourists who arrived in India, in Japan and Delhi, we could receive the sample in frozen condition, and we could use the viro cell culture and isolate the virus. So once we had this virus, immediately we developed different assays which was utilized for the not only vaccine but also for other studies. We developed antiviral kind of standard SOPs, how to we can screen the different products. But at the same time, the ideas immediately floated around that we declared that virus is isolated, and we become fifth country in the world to have the live virus isolates. And then Bharat Biotech came forward, talked to ICMR to develop this vaccine. So once they initiated, so in the meantime, this they came up with the vaccine collaboration. We had animal model established in our facility. We have all the know-how, screening methods, and how virus propagate, how many hours it takes, what would be. So there is a lot goes around developing a vaccine. So we developed those know-how, and then we shared these things with the Bharat Biotech, and they use their GMP facility, standard SOPs, and also the knowledge which they use for developing vaccine. They came up with the candidate vaccine, and then we tested in animal models, including hamster, the non-human primates, and later on, we also work with them on phase one, phase two, phase three clinical trials, and we also did efficacy study on variant of concerns. So also it was a very much collaborative efforts with Bharat Biotech, and it's a very good example for a indigenous making vaccine industry and research bodies together to work in a very crisis time, in a very shortest time, one of this own kind of vaccine we could come up. So it both the team from ICMR and from the Bharat Biotech work tirelessly to come up with this product, and we are very thankful to all the regulatory authorities in India who help us to fast track all the approvals without violating or bypassing any rules. But we received everything in time, and later on, as you remember, in November 2021, we also received the approval from WHO, and we become eighth vaccine in the world. So it's a very, very inspiring story for all the researchers who work in biological sciences to do more for the country, to do at fast track and develop more products which can be useful in India.
Yes, ma'am, indeed. And did you also have a role to play in the development of Covax and Covax vaccines?
No, we were not engaged, but we definitely helped my colleague, Dr. Gajan, and and our team helped them is doing some neutralization assay, but we were not involved basically in those projects. But we did help Zydus Cadila for doing their preclinical study in non-human primate to understand how it was performing in monkeys when once they got their vaccine candidate ready. We also did many other studies for finding, it's for example, finding out if equine sera can be utilized for the treatment. We work with the major many trials which was done in clinical trials to understand the vaccine effectiveness. But also after these vaccines were launched and then emergence of variant of concerns happened, then we did studies human sera samples to understand how they were responding to different emerging variant of concern. Constantly we isolated SARS coronavirus, still we are doing, and we keep studying if it is impacting our immunity, our population, and those data actually useful to understand if we are fine with the COVID. And we know, as you know, that we are totally fine now and we are living a very normal life. So that's very great kind of relief and success for scientific community.
Yes, ma'am. So it means you're still working on understanding the mutations SARS-CoV-2 is undergoing.
Yes, yes, we do that. Yeah.
Recently, the World Health Organization classified Mpox as a global health emergency again. So, is Mpox testing going on in India?
So as soon as the 22 Mpox emerged and it was declared as an international health emergency, in India we had developed this assays very quickly, and then we shared these assays with the VRDL facility across the country based on their geographical location. And initially we had 21 laboratory, which was enhanced to the 35 laboratory. And in last with 2 years, they are constantly testing. But with the alert of this new variant, which is more fatal, the further enhancement and regulatory body has emphasized to do the testing and do the genomic sequencing of all the positive sample whenever it come positive in India and inform immediately. So yeah, this is going on with the efforts of Department of Health Research and ICMR and with the support of VRDL laboratory.
And can you please tell us what other research, apart from what you discussed, your lab is involved in?
So recently, I have received additional responsibility as a director in charge of National Institute for One Health, which is a very future aiming projects for the government of India. And as you remember that in July 22, Pradhan Mantri Modi ji declared about making this One Health mission. So we are focusing on now more on the One Health mission activities, preparing the laboratory which will be ready to not only the detect timely but also do the research and development in the future in a more accumulative manners from different sectors on the principle of One Health.
Yes, ma'am. Would you like to share something more about what future projects you'll be working on?
Yeah, so we are working on some of the vaccine and also diagnosis antivirals for these emerging high risk group of pathogens, which hope should see the day of light and can serve in India.
Yes, ma'am. Can you name some of these pathogens for whom you're working on vaccines?
Yeah. So if you remember in 2018 when Nipah outbreak happened, there was a lot of hue cry. So we established a system is called Mobile BSL-3, which was initiative from ICMR, and we as a team could validate that mobile BSL-3 as a containment facility and can take to the outbreak sites, and you can test your samples on KLR, any other remote areas, and we demonstrated the success in 2023 and 24 outbreak. We are working on some of the antivirals for the Nipah. Uh, we are also working on Casanova forest disease and hope to see some more products in future.
Wow, ma'am. That is so inspiring and so interesting. And congratulations once again, ma'am. And thank you so much for joining us.